This method can help answer key questions in the protein interaction field, such as whether protein binds to a small or large molecule directly. The main advantage of this technique is that it is a simple and fast way to detect direct interactions at the atomic level.
Turn on the airflow with the eject command EJ. This will bring the sample up from the magnet. Now place the sample within a spinner on top of the magnet in the opening. Insert with the command IJ. Wait until the sample settles inside the magnet before proceeding. Create a new dataset using the EDC command and load standard proton NMR parameters by selecting the experiment ZGPR. Fill in the name, experiment number, and processed data folder number fields. Select the solvent in the Set Solvent field and click on Execute Get Pro Sol to read standard probe head and solvent-dependent parameters. Lock the sample to the deuterated solvent using the lock command and wait until it is finished sweeping and achieves lock. Correct the resonance frequency of the magnet by tuning the sample using the automatic tuning command ATMA. Monitor the wobble curve until the automatic tuning is complete. Shim the magnetic field using top shim.
Shimming makes adjustment to the magnetic field to achieve uniformity around the sample. It is good practice to store the shim values with the command WSH and read them using RSH before top shim, if using the same or similar samples.